Now we discussed about the mechanism of DNA transfer in bacteria. Mechanism of DNA transfer in bacteria. There are three types or three mechanisms of DNA transfer in bacteria. First, conjugation. First mechanism, conjugation. Second, transformation. Transformation. And third, transduction. Transduction. There are three mechanisms of DNA transfer in bacteria, that is first conjugation, second mechanism transformation, and third transduction. First we discuss about conjugation. Conjugation. It may be a natural process, natural DNA transfer. Conjugation. There are two strains of bacteria, F plus and F minus bacteria. F plus strain and F minus strains of bacteria are there. The F plus strain, bacteria with plasmid. Now we draw the bacterial conjugation. There are two bacterial cells. We already know the F plus strain bacterium with plasmid. Plasmid is an extra chromosomal or extra DNA strand, that is known as plasmid. The bacteria containing plasmid or extra DNA part is known as F plus strain. This is F plus strain and this is F minus strain. This is the normal DNA part, that is the bacteria with circular DNA. DNA circle. This is the normal DNA part of bacteria. But in case of F plus strain, there is an extra chromosomal or DNA part, that is known as plasmid. Plasmid. Plasmid is an extra DNA part that not included in the metabolism of bacterial cell. So we can utilize the plasmid for our genetic studies. This is the conjugation canal. Conjugation canal. The F plus strain, bacteria with a plasmid, that is an extra DNA part containing few genes. This plasmid containing few genes. Genes are the part of DNA. This F plus strain is known as donor. This F plus strain is known as donor bacteria, and this is the recipient bacteria. This is the recipient. F minus is known as recipient bacteria. First the plasmid rewind. And form two strands. Now we can draw the picture. the plasmid that rewind. The two strands are separated. One of the strand of DNA moving through the conjugation canal and enter into F minus. One of these strand is moving through the conjugation canal and enter into F minus strand. That is the enter into recipient bacterium. The both bacterium with a single strand of plasmid. Now both F plus and F minus bacterium with a single strand of plasmid developed their complementary strand. These single strand then develop into double strand. These single strand formed to their complementary strand, that is double stranded structure. The both bacterium with a single strand of plasmid develop their complementary. We discussed about DNA replication in the former session. So you can utilize this part and can study the different steps of DNA replication. The both bacteria with a single strand of plasmid developed their complementary DNA strand and become double stranded plasmid. And become double stranded plasmid. Now this both the F plus and F minus bacterium become positive stranded. The negative stranded bacterium become positive stranded. This is known as bacterial conjugation. Now we can learn the steps. Conjugation, the steps of conjugation. F plus strain bacteria with a plasmid it is the donor bacterium. The F plus bacterial strain is known as donor bacterium. Bacterium, singular bacterium and plural bacteria. First the plasmid rewind to form two single strands. Two single strands. One of these single strand of DNA move through the conjugation canal and enter into F minus strain bacteria. The F minus strain bacterium, that is the recipient bacterium. Then both the bacterium with single strand plasmid of plasmid develop their complementary strand. Develop their complementary strand and become double stranded plasmid. Now we draw the entire picture of bacterial conjugation. Next type of mechanism of DNA transfer, that is transformation. Second, transformation. We take detailed structure and steps of bacterial transformation in our previous session. You can utilize the part in our channel. Then now we discuss about the second mechanism, that is transformation. Here the DNA from one bacterium may integrate into DNA of another bacterium, as in case of Streptococcus pneumoniae. We discussed about the case of transformation in Streptococcus pneumoniae. The transformation is defined as the ability of extracellular DNA to enter a bacterial cell and combine with the bacterial genome. The transformation is defined as the ability of extracellular DNA to enter a bacterial cell and combined with the bacterial genome, that is bacterial DNA. The transformation is defined as the ability of extracellular DNA to enter a bacterial cell and combine with the bacterial genome. Example: virulent Streptococcus DNA enter into non-virulent. Virulent means smooth coated Streptococcus and non-virulent is rough coated, protein coated bacterium of Streptococcus. There are two strains of Streptococcus bacterium, virulent and non-virulent. Virulent Streptococcus is fatal, that cause disease, pneumonia. But non-virulent rough coated bacterium is not a pathogen. By the process of transformation, the virulent Streptococcus DNA enter into non-virulent bacterial cell and change non-virulent bacteria into virulent bacteria. We already discussed about this part in our previous session. Example, virulent Streptococcus DNA. Enter into non-virulent, that is rough coated, non-virulent bacterial cell and change non-virulent to virulent bacteria. Non-virulent to virulent bacteria. The third mechanism, that is transduction. Transduction. Here a bacteriophage is needed. The transduction refers to transfer of DNA from one bacterial cell into another bacterium through the agency, that is a virus known as bacteriophage. We know the structure of bacteriophage. There are two types of bacteriophages with the core material RNA and DNA as the core material. The rough picture of bacteriophage is look like this. This is the core particle, that is DNA. In this case, DNA as the genetic material are the core particle and a protein coat. The core particle and a protein coat are the main part of a virus. Here the DNA is the core particle in bacteriophage. The bacteriophage is an agency in case of transduction. The transduction refers to the transfer of DNA from one bacterial cell into another bacterium through the agency as a virus. Now we draw the rough picture. This is the bacteria, bacterial cell. Here this is the bacteriophage. Bacteriophage. Now here is the bacteriophage. This is the DNA of bacterium. This DNA is the natural part of bacterial phage. Then this is the DNA strand of another bacterium that need to enter into the other bacterial cell. DNA strand of other bacterium. We need to enter this other bacterial strand to this bacterial cell. The transduction refers to transfer of DNA from one bacterial cell. One bacterial cell enter into another bacterium through the agency of a virus, that is a bacteriophage. We draw these pictures. This is the first and then this is the second picture. Here we can see the bacteriophage penetrate and break the wall of the bacterial cell and transfer the two DNAs to the bacterial cell. This is the normal part of bacteriophage DNA, and this is the foreign, that is the foreign DNA of another bacterium that enter into this bacterium and joined to the DNA of this bacterium. The new bacterial DNA enter from virus and join to the bacterial DNA. This process is known as transduction. Transduction refers to transfer of DNA from one bacterial cell into another bacterium, through or using the agency as a virus. That is here bacteriophage. Now we complete this part. There are three processes: conjugation, transformation and transduction. Now complete this part. If you like this video, please like, share and subscribe our channel.